Hey everybody, we have, as you know, a couple of student talks today. So first we will hear from Shenyang, and then we'll take some questions from the audience, and then we'll hear from Miles, and we'll take some questions from the audience. And then also, just as a reminder, the students will hang back after one when the talks finish, or whenever they finish. So whenever the talks end, we'll have students hang back, and we'll just do a feedback session with Shenyang and Miles and tell them all the things we loved about their talks and any ideas and other questions we have. Okay, great. So, first, our first speaker today is Shenyang, who is a second year in the Cognitive Neuroscience admitting program, was also a Duke undergraduate. So was a neuroscience major here at Duke, or neuroscience and math, I think, actually. And is now studying functional networks in the brain related to memory, moral reasoning, a bunch of cool stuff. So we'll get to hear about some of that today. And something you might not know about Shenyang that I actually got to appreciate during Thanksgiving break is that Shenyang is an amazing chef. So Shenyang's also a whiz in the kitchen in addition to in the lab. So take it away, Shenyang, and we're really looking forward to this.
Well, thanks, Greg, for the introduction. But I should say that that rumor about the kitchen really started with Miles. I blame it on him. Anyway, let's talk about how that counterfactual simulation changed the neural representation of regretful autobiographical memories. This is a project I'm currently working on with Felipe in the IMC lab. So before I dive into the data, let me just give you a little background on counterfactual thinking and autobiographical memory. So counterfactual thinking, specifically in the context of autobiographical memory, refers to mentally simulating alternative versions of past events. So an example could be that I fell off my bike yesterday and ended up injuring my knee super badly. For that event, which is a negative event, I can think of a counterfactual event, which is I could have had my backpack to cushion the fall so that I didn't injure my knee super badly. That thought is referred to as a counterfactual thought, and it's what I'm going to talk about in this project. We as humans engage in counterfactual thinking spontaneously, and especially with these kind of negative regretful memories, we engage in these kind of if-only thoughts spontaneously. But we can also have directed counterfactual thoughts, and those thoughts have been found to have therapeutic effects on those memories. So with directed counterfactual thoughts, we have, if we plot the outcome of the event, of the counterfactual outcome on this axis, we have a better outcome or a worse outcome than the actual thing that happened in the past. So an upward counterfactual is a thing that could have happened in a better way than what actually happened in the past, typically evokes feelings of regret or guilt. And a downward counterfactual, which is thinking about what could have happened worse than what actually happened in the past, typically evokes feelings of contentment or relief. Or maybe it's the absence of counterfactual thinking that is actually therapeutic or beneficial for us, because in the past paper, Felipe and some co-authors find that attentive remembering is the best strategy to reduce the negative affect associated with negative autobiographical memory. So there has been quite a lot of past neuroimaging studies looking at the processes associated with both autobiographical memory and also counterfactual thinking. And they found that the default network, not surprisingly, is involved in both processes, but very few have looked at the related neural representations of autobiographical memory or counterfactual thinking, presumably because that both of them are super unique and multifarious. So the questions I'm going to ask here, first of them is that what brain regions represent details from negative autobiographical memories? Second question, what brain regions are sensitive to counterfactual simulations of those negative autobiographical memories? And the third question would be, how does simulating a counterfactual alternative to negative autobiographical event change its neural representations? Now let's talk about the actual experiment. We recruited younger adults, younger than 30 years of age, right-handed, first language English speaker, and neurologically typical. We had 56 participants. This is a 3-session study. 56 participated in session 1, but a couple of them dropped out. So we ended up having 36 in the final sample. During session 1, which is a behavioral session, each participant generated 45 regretful memories and rated the valence, arousal, levels of detail, and the frequency of remembering those memories for each of them. 1 week later, during session 2, which is an fMRI scanning session, participants engage in, for each of the memory, participants engage in a sequence of trials. So it started with this screen where participants were prompted to remember something from the 45 regretful memories they provided during session 1. So this is a title that they themselves generated during session 1, not studying for final. They think about this event for 12 seconds, and then they press a button when they think that they have already constructed some elaborate image to separate the construction phase and the elaboration phase of the recall of the memory. And then for 2/3 of the trials, which is 30 memories, they also engaged in a counterfactual simulation of this event, not, for example, not studying for final. So they had this if-only prompt, and they also were provided with the direction of the counterfactual thinking. So in this case, the participant should be thinking, if only I have done this, I would do better than whatever happened in the actual event. So that's 2/3 of the trial, which is 30 memories. And on 15 of them, it's a better counterfactual, and on another 15, it will be a worse counterfactual. Again, participants pressed a button to indicate that they have somewhat a vivid image of the counterfactual event. And then 1/3 of the trials, 1/3 of the memories served as a catch trial, for which they didn't engage in counterfactual thinking. They just move on to the next memory, which would be separated by a fixation screen of 1.5 seconds and an active jitter. And then finally, session 3, which is 1 day later after session 2, participants again think about the same 45 memories using the same titles they provided during session 1. In this case, they are only remembering; they're not engaging in any counterfactual thoughts whatsoever. And also for each memory, they provided several ratings that they also did in session 1, so we can monitor the change in those valence or arousal scores. And finally, there is a source memory test, which asks participants for each memory, which condition of counterfactual simulation was the memory assigned to. So that is the whole task. Oh, sorry, I forgot that there is also a button press there. So the analysis I'm doing is looking at this question again: how does counterfactual thinking, better, worse, or the lack thereof, affect the neural representation of those memories? So I'll be looking at the neural representation of each memory for each participant in this phase, and compare that to the second recall in the fMRI scanner to see if there is any representational similarity between those two recalls as a function of the counterfactual simulation condition. Okay, so let's talk about the first question. What brain regions represent details from negative autobiographical memories? To answer this question, as I said before, autobiographical memories are multifarious and unique. We can't directly compare the neural patterns of autobiographical memory recall across the participants. It's not like that we show 15 participants the same image of a cardinal. It's not like that. So that's not possible. However, we do have 2 recalls of the same autobiographical memory from each participant, that is with or without a counterfactual manipulation in between. So we can look at the similarity between the two neural patterns when participants recalled the same memory twice. And to operationalize that, I was looking at, so this is a cluster of voxels in the brain based on fMRI results. We have the neural pattern 1 associated with the first recall of, let's say, not studying for final. For from session 3, we also have a second neural pattern for the same memory. So what we can do is basically compute a Pearson's correlation between those two neural patterns, and then compute the neural pattern similarity score based on that. So after computing the neural pattern similarity, we have to ask, what is the origin of those similarity values? What gave rise to high similarity in neural patterns? The origin of similarity in those values could be either memory specific, because participants are remembering the same event, even though they could vary in some details. So that memory specific contribution should happen for same memory neural pattern similarity. But the origin of similarity could also be memory general, because participants could be familiar with the setup or the instructions, or whatever reasons they might have. And that should contribute to both same memory and other memory neural pattern similarity. So here I'm showing a matrix of neural pattern similarity values for each of the 45 memories across 2 sessions. You can see on the diagonal here, it's the neural pattern similarity for the same memory across 2 sessions. By the way, I'm showing simulated data. This is not actual data, but I'm just showing it so that you can have a visualization of this diagonal being a little bit higher than the others, which would make sense because participants are thinking about the same thing. And then to really compare or to test what regions are representing autobiographical memory details, I would be looking at each row and compare if this diagonal entry, which would be same memory neural pattern similarity, if that value is higher than the average of the others, which would be for this memory 41, its similarity during it's the similarity between this memory 41's session 3 neural pattern and all the other memories session 2 neural patterns. Yeah, hopefully that makes sense. Yes, I said here. So on each row, the diagonal entry should be higher than the average of others. So, oh, by the way, for all of the analysis, I'm using this brain anatomy atlas, which is a parcellation of the brain into 246 RIs. So showing some results. This is just a sample RI, 153, which is the left precuneus. We see that regardless of the counterfactual stimulation condition, whether it's worse, better, or none, the neural pattern similarity, which is on the y-axis, is above 0, which would suggest that for sure there's some memory general similarity across different memories. So that comparison is definitely warranted. And then I compared the neural pattern similarity for the same memory or for across different memories. And we see that for this specific memory, the same memory neural pattern similarity is indeed higher in the no counterfactual case, and also for the worst counterfactual case. It is marginally significant in this better counterfactual case. And I did that for all of the 246 RIs in that atlas. And here's the results. So for a few regions in the lateral and ventral medial PFC, optical gyrus, superior and medial temporal gyrus, inferior parietal labilla, and precuneus, we see a significantly higher neural pattern similarity for 2 recalls of the same memory than for 2 recalls of different memories. So these would be regions that are representing autobiographical memory details. Okay, now we've answered the first question. Let's move on to the second, which is what brain regions are sensitive to counterfactual simulations. So to answer, try to answer that question, because participants are expected to elaborate on their counterfactual thoughts for 2/3 of the memories, their thoughts should, in principle, become more and more different from the original memory as they spend more time on it. So here again, I'm showing the trial structure. During the recall phase, if you remember, participants press the button to separate the construction and the elaboration phase of the recall. Then after a fixation screen of 1.5 seconds, participants entered, for 30 of the memories, participants entered the counterfactual thinking simulation phase, which is also 12 seconds. So what we can do is to separate the counterfactual thinking into 3 segments of 4 seconds each. And then we can compare the neural pattern similarity between the elaboration phase of the recall to each of the 3 segments here. So brain regions that are sensitive to counterfactual thinking should show decreasing neural pattern similarity for later counterfactual segments. So we will be comparing the neural pattern similarity between these 2 blocks and between these 2 blocks. So let me move on to the next. Yeah. Okay, here I'm showing 2 other sample ROIs. This one is the left middle frontal gyrus, and this one is the left rostral hippocampus. For both ROIs, we see that for the neural pattern similarity between recall and the first counterfactual simulation segment is pretty high and much higher than what we have from the second counterfactual simulation segment. In fact, this contrast here is significant for all 246 RIs, which is probably too good to be true. So one reason could be that the first counterfactual segment is only separated from the recall by a fixation screen of 1.5 seconds, which is probably too short. So I decided to only look at the contrast between segment 2 and segment 3. So in here, we can see that for the left middle frontal gyrus, this contrast is significant, where we see higher neural pattern similarity between the recall and the counterfactual segment 3. Between recall and counterfactual segment 2, that is higher than the neural pattern similarity of recall and counterfactual segment 3. That is not true for left rostral hippocampus. So again, after doing all that analysis on all 246 regions, this is a figure of the regions we got showing that counterfactual sensitivity effect I just talked about. And then combining with with regions that showed the memory specific effect I showed earlier in question 1, we have a set of, I think, 6 regions that show both effects. So those regions include regions from the medial PFC, ventrolateral PFC, and the angular gyrus here. Okay, so all that is cool, but finally we have to answer this question: how does simulating a counterfactual alternative to an active autobiographical event change its neural representation? So this part gets a little hand-wavy because I'm still thinking about this part, and I was actually still working on it before the presentation. But yeah, the thought is that any counterfactual effect on the memory could also be either memory specific or memory general, because again, we're using this neural pattern similarity, which requires 2 memories to be correlated. So in this table, we have the same list of memories in their 2 sessions, and then they will have different counterfactual conditions assigned in between. So if the counterfactual thinking effect is specific to each memory, it might visually be looking like this. It's increasing the intensity of each memory. And in that sense, we shouldn't really, in that sense, comparing this not studying for final during recall 2 and traffic accident during session 3 wouldn't really show any effect. But then this counterfactual thinking effect could also be general in the sense that it may just be applying a filter that is similar in color, if you try to think about that visually, to each of those memories that is assigned to the better counterfactual conditions. So in that case, comparing the neural pattern similarity between this not studying for final recall 2 and not studying for final recall 3 would be somewhat similar to the neural pattern similarity between not studying for final recall 2 and too much coffee recall 3. So those are just two possible ways that counterfactual thinking may affect this neural pattern similarity values that we computed. But I'm still trying to think about how we can either combine these two measures to compute a composite measure that's capturing both effects, or maybe tease them apart. So yeah, back again at this correlation matrix, I guess. The memory specific effects would be on the diagonal, and the memory general counterfactual effects would be in each box, where this would be the better counterfactual condition, this would be the worst counterfactual condition, and then finally we have a no counterfactual condition. Yeah, but I'm still trying to think about how we can come up with either one composite measure or several ones to tease apart those memory specific and memory general counterfactual effects. So a little bit of a summary. For the first two questions, we found that regions in the medial PFC, ventrolateral PFC, and left angular gyrus are both memory specific and counterfactual sensitive. With regards to the, I guess, most interesting question of how does simulating a counterfactual alternative to an active autobiographical event change its neural representation, the answer for now is just we'll see. Yeah, if you all have any good suggestions, let me know, put it in the chat, or we can talk afterwards. And I'm also happy to take any questions. Yeah, thanks for listening.
We definitely have some time here for questions. So yeah, go ahead and use raise hand.
So I have a question in the room here. Did your ROIs include subcortical structures? So people will be interested in things like hippocampus or amygdala. Did you look at those structures?
Yeah, those were included, and those didn't show any effect in either of the two. Let me show. Yeah, so this atlas does have a subcortical portion to it, including amygdala, hippocampus, but there was no significant memory specific or counterfactual sensitive effects in them.
I have a question, Shenyang. So the NPS measure is based on just a correlation between the memory at 2 time points, right? Mm-hmm. So you get a lot of defaulty regions, which always sparks suspicion in me. But so I wonder if adjusting the similarity between these two specific memories, more so than the similarity between the memory and all other memories, kind of like the item versus set adjustment that we do with ERF analyses. Do you think that's appropriate here, or do you see my point? So I guess the idea is that it's you want to find the similarity specific to that one memory, right?
Mhm.
And you are looking for the correlation between those, but it might just be that across those two presentations, there's a lot of other things that are also similar. So if
Mhm.
you adjusted for the similarities between, the presentation of a particular memory and a presentation at another time point, for all of the memories that don't matter, right? It gives you the specificity of that memory.
I'm a little confused by your question. I thought you're agreeing with the comparison, this memory specific comparison.
So it is, yeah, I guess I would say I would adjust it by all of the other memories. Yeah, so basically
Mhm.
you would take that identity matrix compared to the average of all of the others, right? So is that what you did, or?
Yeah, that's what I did. Yeah.
Oh, okay, sorry. I just missed that.
Either Zoom or in. All right. Okay. So yeah, if you can stop sharing, Shenyang, and then Miles, Miles, you have control of the screen, right?
I do indeed. Get that up here.
Go ahead and get your slides loaded up.
Sounds good.
Perfect. All right. So, and also, sorry, I put this in the chat, but also for people in the room, there was that feedback form on Qualtrics. If you could please send some notes to Shenyang through that form, and I'll drop the link for Miles also in a moment. So Miles Martinez, our second talk today, also a second-year student in CNAP who was an undergraduate at Brown, and is interested in kind of like two different areas. One is like developing great interpretable models, and the second one is how we learn goal-directed behavior. So we'll hear more about all that today.
Thank you, Greg. All right. So as I've met many of you, just to introduce myself real quick. I'm Miles, and I work with John Pearson. I'm a second year, and I'm going to be talking about modeling tutor-directed dynamics in zebra-finch song learning. There's a lot going on in that title, a lot of things people here may or may not be familiar with. So let's just go through real quick what I'll be telling you all about today. First, we're going to talk a little bit about why we even study birdsong in the first place. Why is that important? We're going to talk about what we already know about birds and their song learning. Then we're going to talk about how we can model and understand this learning problem that birds are solving every day, and specifically what I'm doing currently to understand this learning problem. So, why birds? In cognitive neuroscience, we want to understand how people learn to do things. From the day we're born, specifically, we're constantly learning how to interact with others, how to perform new actions. It's a really interesting learning problem, in my opinion, at least, as Greg said. And so what we want to understand is how people learn to do these new actions. However, the world is uncontrolled. It's hard to understand how we learn these new things by just looking at people in the wild, trying to understand how they learn these new actions. So we typically simplify the tasks for the lab environment. We use model animals and simple tasks so that we can actually come to conclusions about what exactly the brain is doing and how we're adjusting our behaviors. So, we use things like mice. We have them press little levers in the lab. And this is great, and it's led to a lot of really incredible breakthroughs in cognitive neuroscience. It's really increased our understanding of reinforcement learning, of action learning. It's been invaluable. But there's always the question with these model organisms of how well these learning problems translate between the two. How well can we apply learning to press a lever from in response to a drop of juice or some water or some food, to the problem of learning to play a new game with a friend? It's not really clear. in many cases, how we can translate between the two. But luckily, we have this great model organism called the zebra finch, which is a nice in-between between actual human behavior and these simpler tasks. So zebra finches have this very interesting learning problem where they hear a tutor song, a bird teaches them a certain song, and then they just spend the rest of their life learning that song. It's a very complex behavior because vocally these songs are very rich. They have multiple syllables. It's not clear how you get from a bass song to a fully developed song. Yet they're also nice because this behavior, this learning problem, is incredibly constrained at the exact same time. So when zebra finches learn a song, they learn this one song for the rest of their lives. And so they're learning a complex behavior, but they're only learning it once, and it's a continual learning problem where they're just learning it throughout their lives. So this is nice because it's a complex but constrained natural behavior that, due to recent developments in computational methods, also through John's lab, we can start to get a handle on what's going on in this learning problem. So it's a nice kind of in-between for a complex learning problem. So what exactly is this learning problem? We want to know how birds learn to sing their songs. So this is a reinforcement learning problem, but with no obvious extrinsic reward. When birds are learning to sing their songs, it's not like we're standing outside the cage listening to every single song, reaching in, giving them some food whenever they sing something that sounds right. No, that's not how it works. When we do these zebra finch experiments, we expose them to a tutor. The tutor sings their song with them for a couple of days. Then we put the little bird in a box and it just sings to itself for days and days. It doesn't get any food or water reward. It does get food and water. Finches are generally pretty happy, but it doesn't get any reward for singing its songs. And it doesn't have any real social interaction either. It's just by itself, learning to sing, reinforcing actions that are better, and unenforcing actions that are worse. And so this is a really interesting reinforcement learning problem that mirrors a lot of the ones that we solve on a day-to-day basis as well. On the topic of the complexity of the behavior, not really, and behaviors being better or worse, it's not entirely clear which behaviors are better or worse either. In the reinforcement learning tasks I talked about previously, and the behavioral tasks with mice, where they're pressing a lever or not pressing a lever, it's pretty clear that in some situations, it's better to press the lever. In other situations, it's not better to press the lever. However, with finches, we have these really complex songs. So on the top here on the right side here, we have two spectrograms. The top one's from a juvenile zebra finch, and the bottom is from that same zebra finch once it's fully developed its song. And so one of the things we can notice is that the bottom spectrogram is a lot more rich in the spectral domain. There are a lot more vocalizations, there's a lot more structure, but it's not entirely clear how you would get from the first spectrogram to the second. It's not clear how the quality of each of these individual vocalizations is determined. How does the finch know that in this juvenile stage, it's far from where it should be behaviorally? And how does it move from that farther location in behavioral space to its eventual goal? So the more specific question that we're interested in is, how do birds tell that certain behaviors are better than others and then reinforce those behaviors? So there's been a lot of work done on characterizing this learning problem previously, which is really nice. There's been some really beautiful stuff. So just to go into that a little bit, this problem has been characterized very generally, which is really great. And so there's this paper recently by Colmorgan that demonstrated that these syllables become very distinct as the bird ages. So on the plot here, this is a t-SNE colored by what day the bird was when it produced a syllable. And what I want you to notice is that the darker colors are clustered closer together. These are the younger vocalizations. And as the bird ages, those lighter vocalizations spread further and further apart. So the syllables are becoming more and more distinct in a structured way. So the birds are moving towards some sort of behavioral goal. And this is true if you just plot the t-SNE on a day-by-day basis. That's what's shown right here on the bottom. It's actually a pretty rapid progression of these syllables separating into distinct clusters. So the syllables become more distinct as the bird is learning. However, one thing I also want you to notice is that all of the syllables produced on a given day are not all equal to each other. So going back to this top plot right here, figure A, there's a little cluster of these black plus lines and then a white arrow right here. And so looking at this white arrow, what they've done is they've picked a day, just a vocalization from a given day. This is from day 59. And they've looked at which vocalizations look closest to that in terms of the spectrogram. And so what you'll see is that most of the vocalizations in this plot right here at the bottom are centered around that 59-day mark, which is when that vocalization came from. However, there's a distribution of which vocalizations are closest. Some vocalizations that are close to it are from days in the future, and some vocalizations that are close to it are from days in the past. And so assuming that this is kind of the average vocalization from that day, we can think of these vocalizations from the future as worse vocalizations on those future days, because they look more like something that the bird produced in the past. Whereas these are vocalizations that were better for those previous days, because they look more like something that the bird produced in the future. And so thinking about this quality of vocalization in terms of what they look, how syllables look like what the bird will eventually produce, there are actually some pretty interesting patterns to what ends up being learned and what ends up being consolidated based on what looks better and what looks worse. So this other paper, this direct report paper, looked at how these syllables consolidate and found some interesting overnight consolidation patterns. And so on the top here, this is just looking at how much the syllables change over different time periods. And what I really want you to notice is that the biggest changes in vocalization and how the syllables look come from overnight. So that's those red bars there. And specifically, you would think that this would be the syllables becoming better, consolidating more overnight, but actually there's a regression overnight. So this plot here at the bottom is looking at entropy variance, which is essentially a measurement of how vocally rich a given syllable is. And what you can see is that at the end of the day, syllables are kind of consistently vocally rich, but overnight, that vocal richness regresses and they go back to close to their starting point on the previous day. However, again, like I was saying before, there's a pretty distinct pattern to this regression. So plotted here on the right, my apologies, it's a little bit small, is a plot that's looking at exactly this, how syllables regress and how they progress between days. So on the y-axis, you have the difference between the nearest neighbor vocalization of a given syllable, and that is subtracted from the average syllable day of that vocalization. So that's a little bit of a confusing way to say it. Basically what this means is the 50th percentile, these are syllables that look like they're from the exact same day. 95th percentile, that's that black line at the top. These are syllables that look like they're from future days. 5th percentile, these are days that look like they're from previous days. And they're looking at two things here. They're looking at the span, which is the change within day, and they're looking at the shift, which is the change from the end of one day to the start of the next day. And what you can see is that there's a pretty distinct pattern here where syllables that are better, ones that look more like they come from future days, they're consolidated. The gains from the previous day are maintained overnight. Whereas the syllables that look like they came from previous days, the worse vocalizations, these are not strongly consolidated. There is some consolidation still, they do improve, but a lot of what the bird learned on the previous day is not retained. And so the birds are able to differentiate between syllables that look better, that are closer to their behavioral goal, and syllables that are worse, look less like their behavioral goal. So this has been really hard to study in the past because entropy variance and the other features that have been used to study these changes in birdsong and this learning have all been hand engineered, which means that they're inherently biased towards what researchers can already kind of visually pick out. And they might not capture everything that's actually changing in the birdsong. And so what Jack Goffinet and Sam Brudner did, Sam is in the Mooney lab, Jack was with John's lab, he's now in CS. But they used a model called the variational autoencoder to create a low dimensional set of features that were representative of what's actually being learned in the song. So the way that this works, it's similar. You can kind of think of it as PCA or ICA. The latent components that are learned are a little bit different, but it has a similar flavor where you're trying to get at what kind of latent factors are actually governing all of the variance in your data. So what it does is it takes your data, it compresses it to a latent representation, then tries to reconstruct it. And if the reconstructions look good, you can look like the original image, basically. You can kind of confidently say that the information in those images is contained in the latent representation. And so what they found is that song is pretty well described by this low dimensional set of features. So that's really nice. We can actually use these low dimensional features to study how the song is changing over behavioral time. Another cool thing that they did, well, like I just said, we can use these features as a proxy for song, but another cool thing that they did is they split up the song into a finer time scale and used it to train this model. So they call this a shotgun model. It lets us model song on the millisecond level. Basically what this does is it takes a spectrogram, like you see on the top right here, takes about a 20 millisecond chunk of spectrogram, treats it as an image. So that's that red box you see on the top here. Then it slides that window over the entire spectrogram to create a trajectory throughout the latent space. And so let me just show you what that looks like real quick. We start at syllable A, we move over to syllable B across this trajectory, and that little wavy line right there. And it follows a really nice smooth trajectory through the latent space. So that's great. We can kind of look at the millisecond by millisecond variability with in this model using a shotgun encoding or shotgun embedding for our data. So this is where I take over. This is still working with Sam, but what that previous slide and kind of tells us is that latent features tell us where exactly a certain vocalization is, what point in the vocalization we're at. And we can model these latent features to tell us how a song moves during a vocalization, how it moves through the latent space. There's a little bit of math that's incoming here, but we'll go through it together. We'll all be okay. We're fitting this model on the right here, this mathematical model, to our shotgun embedding. So what does this mean? We've got three important things here. We've got Z, which is our position in the latent space. That's that red dot previously. And we're looking at the change in this position in the latent space. This change in the position in the latent space is governed by F, which is directed movement in the latent space. This is basically saying, if we take a step of size dt through time, how far do we go and in what direction? And then we have this third term here. And so this is, you can think of this as diffusion through the latent space. Basically, we're saying with F, we move a certain direction in the latent space, and then we add some extra noise because we might not be super confident in where we should be, or it might be that we want to inject behavioral variability. One thing that's known about zebra finches is that when they're singing by themselves, they tend to inject a lot of behavioral variability. And so this might be important for helping us model how this song actually moves through the latent space. So this is the model that we're using. It's a good time. And what we're going to do is try to learn about learning using this model. So we want to look at how these latent features change over time. And more concretely, how do F and G depend on the bird's age? So previously, what I'm showing you here, F and G, we're just looking at how they change throughout the vocalization. However, we can make them dependent on the bird's age as well, developmental age, and look at how the kind of diffusive force versus directed movement through the latent space depend on how old the bird is, in addition to where it is in the latent space. And so our rationale for doing this is that the shotgun embedding ends up looking pretty different as the bird ages. I'm going to show you a video of it real quick, a video of a UMAP projection of the shotgun embedding real quick. And basically, what I want you to notice is that earlier days in the bird's development are darker colors, later days are lighter colors. And the song essentially consolidates to distinct regions of space as the bird ages. Let's just watch that real quick. So what we can kind of see is that when the bird is young, those earlier purple colors, not sure how well it's coming through over the Wi-Fi. Hopefully it looks okay. But it starts off kind of random and diffuse. And as the bird ages, as the plots get lighter, it consolidates to these distinct strands. And so because of this, what we're expecting is that the song eventually, as the bird ages, becomes more deterministic and has a lower influence of noise on it. So what we expect is that F is going to grow and G is going to shrink as the bird ages. So that's what our hypothesis is. This is what we're working with so far. Concretely, what I've done so far is I've fit F and G using neural networks, and I am going to analyze the magnitude of F and G over time and over latent components. So just look at how big essentially each of these terms are as the bird ages and as it moves throughout this latent space. So for the fitting part, just some preliminary results. It looks pretty good. On each of these plots, we have the real vocalization trajectory in orange and the reconstructed vocal trajectory. These are all in the latent space, by the way, in blue. And on the y-axis, we have different latent components, and on the x-axis, we have time. And our reconstructions end up hugging the real vocalization pretty well. There's just 4 out of 32 latent components, but all of these plots look, they all look the exact same. Nothing exciting going on in any of the other ones. So that's great. Our model does a good job of matching our latent representations. And also, this is very preliminary. I don't want to take this with a very big grain of salt, the largest grain of salt you can possibly find. But if we look at the size of the terms, that's what's plotted on the y-axis in each of these. On the left plot, we have the F term, and on the right plot, we have the G term. It looks like the size of the F term, that directed movement through the latent space, increases over time, whereas the G term doesn't really change. On the x-axis, we have days post-hatch, so that's how old the bird is. As you can see, though, the error bars are really big, and that's in part because I'm not looking currently at how this depends on location in the latent space, because you might have very different sizes of each of these terms based on where you are in the latent space, based on our hypotheses. So I'm going to have to do a bit of a more fine-grained analysis to get at that. So these are the preliminary results so far. Some future steps. I'm going to explicitly force the network to learn the dynamics of song. So these are all neural networks. And what I mean by this is that I'm going to train the autoencoder and the neural networks for the model that I'm using to look at the trajectories jointly. I'm going to train them jointly. And so that way we will hopefully get a better representation of what's going on in the latent space as the bird is singing. That's one thing. I also want to identify how individual components of song change as the bird ages. Part of the whole reason we're using this latent model is so that we can understand how unbiased and unsupervised features contribute to the bird's song. And it might be that each of these individual components, each of these latent features is not equal. Some of them might change more than others. Some might contribute more to the bird's song than others. And so I want to kind of look at how we move through this latent space over time on a component by component basis. And finally, I want to identify how these change based on distance from the tutor song in the same latent space. Part of our whole hypothesis with bird song learning is that the birds know where their tutor song is. They have some sort of model of what their song should look like, and they're trying to match their action to that model. And so it might be that the amount that these terms, the size of these terms are also dependent on how far they are from the tutor song. And if they do depend on that, and if these features are representative of what the bird is actually learning, we should be able to see some differences in the size of these terms based on how close it is to the actual tutor song. So that's all I've got for today. Thank you so much for listening. Here are the people who do the wonderful work. Yeah, that's what I got. Thanks for hanging out. Appreciate it. Any questions?
Yeah, we have a good amount of time here for questions. So if people want to step up to the mic in the front in their room or use raise hand on Zoom. Jaime?
Oh wait. Hi. Yeah, I have a question. So in the I know it's preliminary data that you showed, but do you have any ideas or suggestions about what it means that there's the increased directedness, but not really a change in the diffusion? What would you think that means? Because since you had hypothesized that the diffusion would go down over time.
Yeah, that's a good question. So one of the things that I think this means is that, again, this is just a guess, so take this with the largest grain of salt you possibly can. But what I would expect that this means is that the diffusion that's just along where the bird is supposed to be, so where the tutor song is, that is what shrinks a lot. However, it's pretty rare that you're directly on the tutor song. And so outside of that, you can have a little bit more variance. There's a little bit more room for variance, and that stays pretty consistent. This would also have to do with the variability that the birds actually inject into the song itself when they're singing by themselves for practice. And so that inject variability may stay relatively constant and help them explore this kind of behavioral space a little bit better. Whereas their idea of what they should do, so that would be reflected through this F term, that increases over time. So that would be my guess, that this is kind of reflective of the injected variability that the birds add to their song, whereas the F term is reflective of their idea of where they should be.
That's really interesting. Just to quickly follow up on that, is there something that we should care about this time period that you're studying? Or do you think that these patterns would change if you looked a little bit further along in the development?
Yeah, that's a good question. So I would think that if we look a little bit further along in the development, this F term would kind of end up flattening out. The birds kind of end up with their crystallized song, something that doesn't really change for the rest of their lives when they get to about 120 days post-hatch. And so we should kind of see that level off, whereas the injected variability that they have, from what I've read at least, it stays relatively consistent for these adult birds. And so we shouldn't see any more changes in that. Thanks for the question.
Oh, question in the room.
Hey, Miles, this was great. Thank you. Can you say a little bit more about how you quantify the similarity of two exemplars of a sound?
I can. In what kind of model? Because there are a couple ways to do it depending on how you want to look at the exemplars of the sound. Are you talking about in the Kolmorgen results over here?
Probably before you get to the really detailed learning process, like here, yeah. Like how do you how do you quantify? Like how do you get to those color maps and so forth?
Yeah, that's a really good question. So one of the things that they did here that was really interesting was they treated each of the spectrograms as an image. And so you end up with these, and then they kind of flatten them into vectors. So you end up with, a 128 squared dimensional thing. And then they look at the nearest neighbors by just looking at Euclidean distance between spectrograms. So they subtract one of those vectors from another vector, look at the squared kind of error between the two, and use that as their similarity measure between them. So it's very coarse and high dimensional, but you still end up with these really nice clusters in space.
Thanks. You probably couldn't see me nodding.
No problem. Thank you for the question.
All right, anything else from the room or Zoom? I have just to put you on the spot a little bit here, Miles, unrelated to the exact talk. So Miles is, I think, our first CNAP student to go on to a PhD in engineering.
That's right.
I'm curious just to hear, or maybe for you to share, just about why you chose that path and what you're
Yeah,
excited about.
Yeah, that's a good question. So one of the things I was really interested in undergrad that I got interested in through my previous mentors, one of my previous mentors, he was a postdoc at the time. He's now a data scientist. His name's Tony. But he got me interested in computational modeling. I was doing some stuff with motor learning, and I thought that it was a really cool way to just be able to describe behavior in a pretty precise way. But then I kind of looked into it more in psychology and neuroscience, and I was like, wow, there's a lot of stuff here that's kind of really, really shady mathematically. And I didn't want to be one of those people. I definitely want to do it right, and I thought that I could get a really great education through an engineering department. I could get a really good education through the engineering department to get at that. And I also think that I'll be able to really understand a lot of super cool tools like these VAEs, like the differential equations that I'm working with now, if I continue along this path. That's one of the reasons. Another one of the reasons is that I can, since I'm in CNAP, I can still keep doing neuro research. I don't think I would have done it if I didn't have the ability to continue along this path as well. Like you mentioned, I kind of have those two split interests. So this allows me to indulge both of those pretty well. And finally, it's definitely going to give me options once I graduate for what I want to do. I want to keep my options open. Definitely still looking into academia, but it's a tough world out there. And so this will give me the option to be pretty competitive in industry too, if I end up going with that path.
Cool. Thank you so much. Sorry for the dancing in the background.
Oh, please, no need to apologize for that at all.
All right. I mean, yeah, we have a couple minutes, so I don't know if anyone else in the room, or we can just move on to the student session. So, in 1 or 2 minutes after people clear out of the room, we'll have a feedback session together. Great job, Miles and Shenyang.
Thank you.